Plant development occurs without cell movement. For us, though, cell migration is very important. We see it not only in animal embryonic development, but also in immune responses and cancer metastasis. But how do cells move? Cytoskeletal theory provides one explanation. There are three key components to cell motility. The first is adhesion. A cell cannot move properly if it is slipping across the surface. Next, the cell needs to be able to stretch in the direction it is trying to move, adhering to its new spot. And finally, the cell needs to let go at the back end and contract away. This forward movement is facilitated by a phenomenon known as actin treadmilling. No, not that kind of treadmilling. This is an actin molecule. These molecules assemble into cytoskeletal structures called microfilaments. Initially, the binding of actin monomers is energetically unfavorable. Filament assembly begins with a process called nucleation, or the formation of an actin trimer. Once the trimer has formed, it becomes energetically favorable for additional actin molecules to bind, elongating the filament. The longer the filament gets, the less stiff it is. Short branches provide more rigid structure. Naturally, the subunits assemble head to tail along the filament. So how do we get these branches? There must be a protein complex facilitating a branch point. Two molecules known as ARP2 and ARP3 are strikingly similar to actin in their homology. They come together to form a 7-subunit complex. But the ARP2-3 complex won't interact with the actin filament in this form. It is in the inactive state. An activated WASP molecule binds to an actin protein which also binds to the ARP2-3 complex, strengthening its interaction with the actin strand. The stem has now been nucleated and can continue to grow by polymerization. But why do actin monomers only add to one end of the filament? Microfilaments are polar. Subunits usually add to the plus end and dissociate from the minus end. The plus end is rich in actin ATP, while the minus end is rich in actin ADP. Addition to the plus end of the filament is promoted by profilin. Profilin is an adenine exchange factor. It helps actin exchange a molecule of ADP for a molecule of ATP. The newly formed actin ATP can now bind to the plus end of the filament. Meanwhile, at the minus end of the filament, cofilin promotes microfilament disassembly. Cofilin binds to the side of the actin ADP filament and depolymerizes it. These free subunits can then be bound to profilin, readying them for addition to the plus end of the microfilament. And that's all for actin treadmilling.